Part 2, Chapter 4. If you can figure out two things about the epithelial tissue slides that you're looking at, you're like 80% of the way to knowing all of the epithelial tissues, okay? It's kind of like having a first name and a last name. You've got the shape of the superficial cells and the number of layers. If you can tell me that, you're good as far as knowing which type of epithelial tissue you're looking at. So let's go through the shape of the cells first. We've got three options when it comes to shape. There's squamous, there's cuboidal, and there's columnar. Squamous are squished cells. They look like pancakes, okay? So if you can remember squished, you should hopefully key in on the word squamous. Then you've got cuboidal. Now, cuboidal cells are about the same height as they are width, okay? So they're not going to look like a perfect square. They're probably going to look something like this or something like this, but they are about the same height as they are width. So if you were to look at them in 3 dimensions, they would look like a cube, okay? So that's the second shape we're looking at. The third shape we're looking at is columnar. So think of a column for a second. Roman Colosseum, right? The column. Columns tend to be a lot taller than they are wide. That's normal. So when we talk about the shape of a cell and you see a cell that is a lot taller than it is wide, you know that it belongs in the columnar category. Again, if you know the shape, you've got half of the name already. You don't even have to memorize the slides. If you know shape and you know the other half of this equation, you already know what to call the slide. So let's talk about the other half of the equation. The number of layers of cells, okay? So with that, we only have basically 3 options, okay? But even with the third option, that one's a little bit harder. So you will have 1 layer of cells, or you will have more than 1 layer of cells. This is that 80% I was talking about earlier. If you have 1 layer of cells, it is called simple. So your first name, in this case, 1 layer of cells, it would be simple. Second name, the last name for this particular picture, what's the shape? Are they cuboidal? Are they columnar? Or are they squamous? Are they squished? They're squished, right? So this is simple squamous. Ta-da! I'm done. I know the number of layers. I know the shape of the cell. First name, last name, done. Now, if you look down here at the second picture, I've got a lot of layers of cells, right? Although most of them kind of look squished. You can see that up here at the top, right? So when I have more than 1 layer of cells, the first name is going to be stratified. Okay, so instead of simple, which means 1 layer, we say stratified. That's my first name. Now, what's the shape of those cells? Are they taller than they are wide? Are they about the same height as width? Or are they squished? They're squished, right? These little dudes up here, they all look like almonds, so they're all squished. So this is stratified. Squamous. Again, first name, last name. If you know the number of layers and you know the shape of the cell, you know what you're looking at. Now, something that is kind of a little bit of a fly in the ointment as far as being able to be 80% of the way there is this one, okay? This one. Pseudostratified. Pseudo means fake, okay? So what did stratified mean? Stratified meant multiple layers. So if it's pseudo-stratified, I'm saying fake layers, okay? So if you look at the example over here on the left, whoops. If you look at the example over here, it does kind of look like there are multiple layers of cells, but this is the key to pseudostratified. There is only one layer of cells. And the reason that we know that is because all of these cells are touching the basement membrane. Even if it's just in one spot, one single solitary spot, all of these cells in some way, shape, or form come down and touch the basement membrane. So it is literally only one layer of cells. With pseudostratified, you actually, basically, you think it's more than one layer because the cells are all kinds of funky shapes, but it's just one layer. Now, for my class, for my lab, you always know pseudostratified columnar because it has cilia at the top, just like this guy looks like he's got little fur up here. So pseudostratified columnar, ciliated columnar, because of the cilia, are going to kind of stand out as a slide for histology. All right. So, the functional characteristics. The structure and organization of the cells within each epithelial type represent or reflect their function. So, remember, we've got the option of having one layer of cells. We've got the option of having multiple layers of cells. We're going to talk about what the layers mean, what the shapes mean, what the free surfaces look like, and what those could mean. And then we're going to talk about those special cell-to-cell connections that I was talking about earlier with the epithelial tissue. So, layers. If it's simple, it's one layer, okay? It means you're going to be moving stuff, either from the cells over or through the cells. So you've got things like gas diffusion. Gas diffusion happens in the lungs. We need to be able to take oxygen from the atmosphere into our blood. And I can't have a huge, ginormous wall between a gas and where my blood is, right? So I have simple squamous in my lungs. And that means that if this has to get past this, it's not really a huge, thick wall that I have to get past. And you can see that. It's just this little thin thing right there, okay? Sorry, my phone is blinking and it's bugging me. Another thing, if we've got simple, we can see filtration. Think about the coffee filter. The coffee filter is not as thick as, let's say, a quilted blanket, right? It's very thin so that the liquid will go through, but the coffee grinds, won't. So we use, again, just one layer of cells to help us do this function, filter. Secretion. We secrete products. Let's go back to our oil and our sweat glands in our skin. Those are actually tubes that are a simple layer of cells, but those cells that line those tubes, so here's my tube with my cells in it. Just go with me on my delusion that I'm an artist. These cells put their product into this lumen. Remember I told you lumen just meant hole? So that is actually one of the functions of a simple layer. And then finally, we've got absorbing. In the intestines, we have a single layer of columnar cells. So instead of having cuboidal cells in this tube that I just drew a minute ago, we have columnar cells. Again, pretend I'm an artist. And when food comes through here, these cells actually absorb those nutrients and bring them into the body. Okay? So simple. Simple means that I'm either going to be putting something into the space in front of me, or I'm going to be bringing stuff the opposite direction. I'm going to bring stuff past me. If I'm stratified, remember, stratified means more than one layer. My main function is going to be protection, always. That's just the way it works. Now, shape. If I have flat, thin cells, if I've got these squamous-like cells like I have up here, it's going to be diffusion or it's going to be filtration. I need to get things through and I need to do it really easily. Cube or columnar, usually those are going to be involved in secretion and absorption. Why? Because their job is a bit more complicated. It means that I have to be able to make the machinery to do those jobs. Ultimately, what that means is I've got to have a bigger building. So my cells get thicker as my job gets more complex. I always discuss in my class, let's say that I have a crocheting business. A crocheting business is me, let's say, making amigurumi. I make little animals and I crochet. I can do that in my bedroom. So my bedroom can be my workspace. I can have, skeins of yarn and I can have crochet hooks. That bedroom is big enough to accommodate that. So it's a simple business. It's not that complicated. Now let's say I want to build cars. Can I do that in my bedroom? No. Why? Because building cars is going to involve me having bigger machinery that won't fit in a bedroom. Maybe like on campus, I can use this building and, have a car factory here where I've got the big machines to lift the heavy parts of the cars and all of that. But I would need more room to accommodate that machinery. It's the same concept. With cube and columnar shaped cells, the cells are bigger because their jobs are more complicated, which means internally I've got to house more complex things. Okay, let's talk about surfaces. The free surface can either have a smooth edge, can be smooth like glass. In this case, we're talking about reducing friction. Your blood vessels, we want those to be smooth like glass so that your blood flows through them without any issue. We know what happens when you don't have that. You get, blockages and you get plaques and you get all kinds of issues. We know that that isn't how it's supposed to be. It's supposed to be a smooth edge. You also have the possibility of having microvilli. So microvilli, if I have a cell and it's that big, but I want this part of my cell to have a lot more surface, doing this with my cell isn't really a good idea because that would mean it would take up a whole lot more space. So what can I do with that edge at the top? So normally that edge looks like this. And I want more of that surface right there. What can I do? Well, I can do this with that surface. That, technically, if I unwind that accordion, would probably be about that long. So did I increase the amount of surface? Yes. Did I take up more space? No. So that gave me a lot more surface without necessarily taking up more space in my body. That type of surface is a microvilli. Okay? Microvilli increase surface area without increasing space. Now, where would I maybe want to have extra surface? One of the places is the small intestine, because what's the job of my small intestine? It's to absorb nutrients, right? So if I'm trying to absorb nutrients, I need as much surface as I can. I need as much opportunity as I possibly can. Not only does having these guys give me surface, it also kind of makes speed bumps so that things kind of have to slow down as they're moving. Okay, now ciliated cells. Ciliated is different than microvilli. So again, let's draw a cell. So here's my cell. Ciliated means that I've got these little hairs that are sticking out on top of my cell. They're not microvilli. They are not about extending surface area and giving me more surface. They're actually about moving things along the surface of my cells. Okay. If you know anything about bacteria and you had any type of basic bio where they talked about the cilia or the flagella causing the cell to swim around, our cells don't do that. Our cells don't move. Once they are in place in our body, they really don't go anywhere. So our toe cell isn't going to come up to our intestine and go, please, sir, may I have some food? It doesn't work that way. So having cilia on some of our cells will not make them move. What it does do is it allows these cells to use the flagella to move things across its surface. So the example I always give, I have 3 older brothers, right? I have 3 older brothers, and if I ever wanted anything, I had to fight for it. My oldest older brother is 10 years older than me. So when I was 5 years old, he was 15, okay? So long story short, Mom used to buy us pixie sticks. Everybody knows the pixie stick, right? It's a straw with a whole bunch of sugar in it, usually flavored, right? If Mom gave us those and I didn't immediately eat that, I lost it. It was gone. So I would always rip it open really, really quick and go, yeah. Then what happened? Because instead of putting it in my mouth and swallowing it, I kind of breathed it, which was not a good idea. Okay, so why do I mention this? Well, that breathing tube that I'm talking about is lined with ciliated cells. Okay, so here's my breathing tube. Here are the cilia that line it. When I inhaled pixie stick, okay, what happened, let me see, give me a second. I ended up with pixie stick dust all the way down into my body. That is a horrible color. Give me another second. Let's try this one. Nope, that one's not any better. Try again. Let's go with that maybe. Yes, better. So when you initially breathe this in, goodness, sorry. When you initially breathe this in, you end up with pixie stick dust going all the way down, okay? Not necessarily all the way down to your lungs, but quite a bit of a ways. And if you've ever paid attention, you kind of do the right after, and then you're like, I'm okay, right? But 20 minutes later, you've got the tickle in the back of your throat, and you go, no, I'm okay. Right? And then 20 minutes after that, you get that tickle in. I'm okay. But like for an hour, you're still going, right? Okay. So what has happened? Let's say this is the back of your mouth, right? When you first choke, this guy right here is what you're going and getting out. And you get him out. Well, these little hairs here kind of act like a crowd at a concert, crowd surfing. So if you're crowd surfing at a concert, Are the people under you moving with you above their heads as you move across the crowd? There's this group of people holding your legs and just walking with you? No. What do? They pass you along, right? So the people underneath you are staying still, and all they're doing is they're passing you along to other people down the way. So this actually comes up next because these little hairs, these little cilia, are crowd surfing it up to the top, and then in 20 minutes you go and you need to get rid of it. So you get rid of it. Well, by that point, this one has moved its way up to here. So in 20 minutes, it moves its way up to here, and you go and you get rid of it. So we call this an escalator, actually, because it moves these particles out. But again, the cilia are moving, but they're moving stuff across their surface, like the crowd in the rock concert. They're not moving the cells themselves. So when it comes to the cilia or the ciliated surface, it's moving things along the surface. And again, the perfect example is the respiratory lining. So let me erase that. The last one, folded. If you have a surface that is folded and you fill that, let's say, space. So I've got something that looks like this, a deflated balloon, right? And then I fill the balloon with air. This straightens out, right? It doesn't stay all floppy. You don't have a balloon that looks floppy like that. I'm pretty sure that if you bought balloons like that, you'd go take them back and go, these aren't what I asked for. So when you have a folded cell surface, something that looks kind of like this, and you fill that surface, it actually allows it to kind of fill up or bulge out, and it'll smooth out, but when it's empty, it goes back to looking like this. The place that we see this folded surface is actually in the bladder. When your bladder empties, it looks like the deflated balloon. When your bladder fills, it looks like the inflated balloon. But the surface itself has this folded look to it so that when it does compress down, it's not like you lose that surface. It's still there, it's just folded up. Now let's talk cell-to-cell connections. These are either going to hold the cells to each other or the cells to the basement membrane. So I'm either holding on to my neighbor or I'm holding on to the thing that's keeping me attached to the body. They have three functions. They can bind the cells to each other. They can actually form a permanent barrier, or they can provide a mechanism for cells to communicate with other cells, intercellular communication. Remember, inter is in between. Okay? We have four types: desmosomes, hemidesmosomes, tight junctions, and gap junctions. So let's look at those. Desmosomes. Let me get a better color than that. Go with red. This guy right here. Desmosomes have these disc-shaped structures and these filaments that go through, that anchor through those disc-shaped glycoproteins. And they help to bind the cells to each other. Basically, they're binding you to your neighboring cell. So if you look, here's cell 1, here's cell 2. This is keeping them attached to each other. Okay? Hemidesmosomes are the second one we're going to talk about. Hemi means half. Okay? So this has this disc-shaped structure and then these filaments, right? But over here, there's a second disc-shaped structure with these filaments over here. If you look at the bottom, though, There's a disc-shaped structure and there's filaments, but there's not another one over here on this other side. Instead, it's the basement membrane. Remember, basement membrane is the double-sided sticky tape that helps to hold the cells above it to whatever's below it. So hemidesmosomes bind your cells to the basement membrane. They anchor them down. Tight junctions. That's this little dude up here. Tight junctions do exactly what they sound like. They bind the cells so tightly together that nothing can go in between the cells, okay? That should be a T. The tight junction itself is what's forming that tight seal. If you look, you can see that this is like held together like nobody's business, right? But right next to it, you also have this little thing here called the adhesion belt. The adhesion belt is kind of like having weak glue right next to the strong glue. It's not necessarily going to be as good as the tight junction, but it adds a little bit of stability to that tight junction. Tight junctions are usually found in the lining of things that we don't want stuff leaking from. Your stomach contains hydrochloric acid. I don't know about you, but I'm thinking that if that hydrochloric acid gets out of my stomach into my internal organs, hydrochloric acid doesn't have a personality. It's not going to go, oh no, that's my own liver. I'm not going to burn that. No, it'll burn it. So having tight junctions in the lining of the stomach ensures that that acid stays there, which is what we want. Then finally, we have gap junctions. Gap junctions make a gap. They literally make a space in between cells. It's small contact points between cells, and it allows for intercellular communication. So I'm going to blow this up. See these little flowers here? Okay. If you look, see that tube going from this cell to this cell? If I have chemicals that are a form of communication in this cell, I can literally just use this tube to get them into my neighbor cell. I don't have to worry about I have to go out of the cell and start a chain reaction so that I can get the cell to understand what I'm trying to tell it. No, I literally have something in here and I can just pass it along into the next cell. It would be like being in an enclosed space in, let's say, the school and having a pipe into the room next door. And let's say that in my room, I have a sink. In their room, they have a toilet. Well, I can collect waste here, put it through the pipe, and they can put it through their toilet. And I've got clean water, so I can fill buckets and give them to them with clean water. We can exchange back and forth between the two rooms. These cells can exchange back and forth between each other because of these gap junctions. Okay, glands. These are secretory organs, meaning that they secrete a product. Many are composed of epithelium, like I said before, but they also usually have supporting endocrine tissue. Supporting connective tissue. I was reading ahead, sorry. We have two types of glands. We have endocrine glands and we have exocrine glands. And the name kind of gives it away. Both of them make a product. Obviously, they're glands, they make a product. But endocrine glands make products that get secreted into the blood. So hormones are an example of this. You don't go, I'm sweating testosterone today. No, that doesn't happen because it's only secreted into your body, into your bloodstream. Exocrine glands, however, are going to be glands that secrete a product onto a surface. So your sweat gland is a good example of that. Your oil glands on your skin are good examples of this. If you look over here, you'll notice that I've got kind of these simple tubular glands. They look like a test tube. And then I've got this little dude over here that looks more like a grape, right? These are called acinar glands. And the thing is, these simple tubular glands can be really, really long. And when they get really long, they tend to kind of look like a hose, like somebody took a hose and dropped it. So there's a bundle of hose at the bottom and then this one piece of tube that's coming up. Your sweat glands in your skin look like that. Technically, it is still a tubular gland. It's just really excessively long. So it ends up coiled, kind of like what you see here. Now, with the acinar glands, you can actually have these branched acinar glands so that instead of it just looking like one grape, you almost have bundles of grapes, like what you see down here. Okay? And they're called compound glands when you do that. All right. So, how do exocrine glands work? How do we get product into that lumen, into that tube? We've got merocrine glands, we've got apocrine glands, and we've got holocrine glands. Okay? The product that they make and how they get it into that lumen, into that space, is how I'm classifying them. So, merocrine glands, they produce their secretions and then they exocytose it into the lumen until it works its way to the top. This is going to be the most common type of gland that we have. Most of our glands are going to be merocrine, where I make a product, I exocytose it, and it goes into this duct. Okay? Apocrine glands are different because literally with apocrine glands, I am pinching parts of my cell off and it's becoming the product. Now, normally, I don't want you to think you've got chunk of cell coming out of this gland because that isn't it either. Normally, these things rupture and eventually whatever is in this bubble ends up being part of the excretion that's happening with the apocrine gland. And finally, we have the holocrine gland. Holocrine glands take the whole cell off of this lining and it becomes part of the product. Again, does the whole cell come out of the top of the duct? No. Honestly, most of the time, just the trip is enough to rupture it. So the guts of the cell are what's going to be coming out of that. Okay.